Please solve this interesting question. A molecule of amylopectin contains 2000 glucose residue and branch present at every 25th residue. Total how many reducing ends are present in this molecule? So before going this question, let's compare starch and glycose. Starch, and we will see what is amylopectin, and we will compare starch and glycogen also. Starch, you all know, it's stored food in plants. Our starch is stored food in plant, isn't it? Plant stored food. Glycogen is stored food in animals. Yes, animals. Starch is mixture of two kind of compound. One is amylose, another one is amylopectin. But glycogen, if you'll see, glycogen is not mixture, right? Here amylose is 20% approximately, and amylopectin is 80%. Remaining one is 80%. If you'll see monosaccharide, because starch and glycogen both are polysaccharide, you will see monosaccharide, they are made by alpha glucose. Either glycogen or starch, I mean amylose and amylopectin, are made by alpha glucose. Yes. Now, particularly if you see this amylose have alpha glucose linked by alpha 1,4 glycosidic bond. Yes, they are linked by alpha 1,4 glycosidic bond. In amylopectin, between two alpha glucose alpha 1,4 glycosidic bond present, but they have branching also at every 25th glucose residue, and this branching is alpha 1,6 glycosidic bond. This is big difference between amylose and amylopectin, that amylopectin have branching at every 25th residue, but here branching absent. In our amylose, branching absent. Yes, branching absent. Now come to glycogen. In glycogen, alpha glucose are present, right? And here alpha 1,4 glycosidic bond present. Then alpha 1,6 branching also present here. This alpha 1,6 glycosidic bond branching present at every 12th glucose. 12th glucose. Now you tell me that out of this starch and glycogen, who have more branching? Here branching is present at 25th glucose, here branching present at every 12th glucose, so obviously glycogen have more branching. Yes, glycogen have more branching, right? Is it or not? Yes. Now the amylopectin, I mean starch, when starch will mix with iodine, we will get blue color compound. Starch will mix with iodine, we will get blue color complex. But color complex. But when you will mix glycogen with iodine, then we will get red color complex. Glycogen plus iodine, red color complex we get. Now come back to question. We compared amylose, amylopectin. Amylose is unbranched actually, right? And it's helical, you know, because of absence of branching, it can be easily twisted, and it's actually helical. Okay, amylopectin or glycogen are not helical, right? Because of presence of its helical structure only, because of amylose only, our starch is blue, forming blue color complex. That kind of helix is absent in glycogen, that's why they will not form blue color complex. Now come back to question. This is, suppose this is one chain of glucose. First carbon, yes, second, first carbon, second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon. This is another alpha glucose. First, second, third, fourth, fifth, sixth carbon, right? And this is glycosidic bond present between first and fourth carbon, alpha 1,4 glycosidic bond. Next also about this alpha 1,4 glycosidic bond only present in next glucose also, right? But if even n number of glucose is added, last glucose will be free. I mean the last glucose, if you'll see there, first carbon is free, first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and sixth carbon. I just want to say that even we have a long chain of glucose, and between two glucose alpha 1,4 glycosidic bond present. So if you'll consider last glucose, in that last glucose first carbon is free or not, they are not forming any kind of glycosidic bond, isn't it? And you all know, if glucose have free aldehyde or ketone group, we will call that anomery group. So if anomery group is free, we will call it that in reducing it. Okay, and you see first carbon is engaged in formation of glycosidic bond here. That means first or second or third or n number except last one, everyone first carbon is involved in formation of glycosidic bond, so we will call it non-reducing. That end will, first end will be non-reducing, last end will be reducing. Now either you are taking glycogen or you are taking amylopectin, they have branching. And which kind of branching they have? Alpha 1,6 branching they have. Like see here we have sixth carbon, right? This sixth carbon will react with the first carbon, right? Like you see first carbon of any other glucose, and then they form alpha 1,6 glycosidic bond. So you see here student, at the branching first carbon is engaged in formation of alpha 1,6 glycosidic bond or not, so there is no question of reducing end here. So either you take 2,000 glucose, or you take 5,000 glucose, or take 30,000 glucose, and they are connected by alpha 1,4 glycosidic bond, and branching alpha 1,6, they have only one reducing end. Okay, they have only one reducing end. At branching, no reducing end. Remaining we'll see in next video. Bye.